Park Cell Supply Center here at the Diabetes Research Institute, where we're going to speak to Dr. Dominguez Bendala, Head of Stem Cell Development Program here at the DRI. Come with us to his lab where they're working on some really exciting things to work toward curing diabetes and creating an unlimited supply of insulin-producing cells. So step aside. Hi, Doctor.
Hi, how are you? So nice to see you.
Thank you so much for having us.
Please, my pleasure. Come in.
All right, well, we have a lot of our supporters on live right now on Facebook, and we're just going to talk a little bit about what you're doing here, all this exciting stuff. We want to start with the fact that your team is pursuing several strategies to create an unlimited supply of insulin-producing cells. Why is cell supply so important in the path toward a diabetes cure?
Right. Well, type 1 diabetes, as we all know, is an autoimmune disease. And as a result of this disease, the cells that make insulin within the pancreas are lost. So it's imperative that we replace those cells. Any cure has to tackle that issue. We need to replenish the beta cell mass that has been lost through the autoimmune process. We've been doing this over the years by islet transplantation, and in fact the DRI is one of the foremost centers in the world that has been pioneering this technique. There's only so many patients that we can transplant using this approach. There's about 3 million people with type 1 diabetes just in the United States, and we can transplant only a handful of patients every year. So that's why it's so important to get more cells that can satisfy the demand for the treatment.
Clearly, there's a great need for these cells, and I know you guys are doing a lot to tackle that. What are some specific strategies that you are pursuing currently?
Well, there are many. We are pursuing many projects, but one of the most promising ones is actually something that we started working on with my colleague Ricardo Pastori 5 years ago. And it's based on the fact that every time we do islet transplantation, we extract the islets, which are only 1% of the pancreas, and then throw away the rest of the organ. We have no use for the rest of the organ, which is exocrine tissue. That's tissue that's involved in making digestive juices, but it doesn't make insulin. We thought it was a waste and started to look for ways to recycle that part of the tissue that we have no use for. So we came up with this molecule called BMP7, or bone morphogenetic protein 7. It's an agent that is already approved by the FDA for the treatment of autoimmune conditions, and we found that when we expose exocrine tissue of the pancreas to BMP7, new islets sprout and form. So we have potentially the means to reconvert or recycle 98% of the pancreas into new islets. Actually, this is a picture that I always show. This is one of the new islets that we get when we expose the exocrine tissue to BMP7. And what you see there in green is insulin. This tissue normally doesn't express insulin, but when exposed to BMP7, new islets, as I say, grow throughout the plate. So we now have two potential means to exploit this finding. One of them is to get from one organ multiple patients, transplant multiple patients. But the other is that we found that the cells that respond to BMP7 in the tissue are actually stem cells. And that was a novel discovery. Now we have also potentially the means to stimulate those cells within the body of the patient and recreate those cells without the need for transplantation. And that's something that we are very actively exploring right now.
Wow, this is really, really exciting stuff. And you say, you know, you launched the stem cell research program here at the DRI more than a decade ago,
yeah.
And a lot of exciting things have clearly been happening since. How has this area of research evolved since you began, and what are your hopes for this research in the near future?
Well, it's been a long ride, and we have seen things that were very promising at one point and ended up being dead ends, and that's the nature of research. But the good thing about working on this for so long is that eventually the dust settles, and now I think that we have a clear view of the things that have the highest chances to get to the clinic. So let me give you an example. Embryonic stem cells. Just a few years ago, to think about the use of embryonic stem cells for the clinical treatment of diabetes was a very distant possibility in the future, and now we are witnessing it. It's happening right now. We are witnessing clinical trials using human embryonic stem cells. So I think that the future is possibly going towards inducing the regeneration of cells within the body, circumventing or bypassing transplantation altogether. I think that before we get a cure, we'll see treatments or therapies that have the potential to completely change, dramatically change the quality of life of people living with type 1 diabetes. But we are very cognizant of the fact that the cure is the ultimate goal. Nothing short of a cure is what these people who live with type 1 diabetes need, and we are not going to stop at anything before we get it. So that's what we are exploring.
Well, thank you so much for your time and your expertise, and obviously the great work that you're doing. It's clear that what's happening now in your lab is definitely pushing forward and closer, getting us closer to developing a cure for type 1 diabetes. So we also would like everyone to know that there is a way you can help Dr. Juan Dominguez Bendala's team multiply cell supply now. Join our $100,000 match challenge. Every dollar you donate between now and December 31st will not only go toward funding this critical area of research, your gift will double thanks to the generous match by the Fred and Mabel R. Parks Foundation. We will post a link below shortly for those of you who may not get a chance to see this. And thank you so much again to everyone, to Dr. Juan Dominguez Bendala, and to all of you out there who tuned in and are supporting our important work toward curing this disease.